This is a presentation on human immunity by Rose Rogers. Section 1. Non-specific defences against disease, including the first and second lines of defence, barriers to infection, inflammation, fever and role of macrophages. General concepts. Most viral infections are limited to the defences that are non-specific or specific. Non-specific act quicker than specific defences because they're always in place. Automatic barriers, non-specific inhibitors and phagocytic cells. Others can happen by infection, and symptoms include fever and inflammation. Automatic barriers. Barriers are found in the body's surfaces, skin and mucosa, within the body's endothelial and basement membranes. These can be effective by preventing viruses spread, and they can be breached by large numbers of viruses, including trauma and replication of viruses in endothelial cells and transportation of the virus and leukocytes. Body fluids and tissues contain soluble viral inhibitors, preventing viral attachment of some directly inactive viruses. However, others interact intercellularly, but these inhibitors can be overwhelmed by the virus. This is a picture of a phagocyte, and it just goes on to say some phagocytes are at different levels: polymorph, leukocytes, macrophages. The effect of phagocytosis can be virus inactivation. Fever. Viruses are reduced by even the modest rise in temperature during an infection. Fever is started when several endogenous pyrogens, like interleukins and tumor necrosis factor. Inflammation. Inflammation inhibits the viral replication through elevated temperature, reduced oxygen tension, metabolic alterations, and acid productions. The effects of these mechanisms are sometimes addictive. Here's a picture of an inflammation there. Interviral interface and interferon. Viral interference happens when infection by the viruses makes cells resistant to the same or other infecting viruses. They're usually mediated by newly induced host cells, proteins made as the interferon systems. This then binds to the cell and makes them block various stages of the viral replication. And they also stop growth of some normal tumor cells, moderates immune response, affects different cells, differentiation cells. These the three types of interferon: alpha, beta, and gamma. Alpha are produced mainly in certain leukocytes, dendritic cells, macrophages, and B cells. Beta interferon by epithelial cells and fibroblasts, and gamma by T cells, natural killer cells. And this is a picture of interferon and interference and interferon. Here's another picture of interferon and a related interferon, alpha interferon. Section 2. Third line of defense can be acquired adaptive or specific immunity. Here's a picture of the third line of defense. Physical barriers is the first line of defence. This is divided into two categories, physical and chemical. These barriers are non-specific in residence. Physical barriers, which include the skin and mucosa in the digestive and respiratory tracts, help get rid of pathogens and prevent tissue infections in the blood. These parts are secreted by the skin and mucosa, such as sweat, saliva, tears, and they help provide a basic barrier against invading pathogens. And they have a picture of a barrier, physical barrier of immunity. Non-specific innate response. Pathogens that do cross the physical barrier and are next encountered by the second line of defence, which is the immune response, mostly involves immune cells and proteins, and non-specifically recognise and kill pathogens that enter the body. Phagocytosis is vital. An innate immune system that utilises the special types of immune cells called phagocytes. There we go. And phagocytes recognise and bind pathogens that then use the plasma membrane to surround and engulf the pathogens inside the cell. As a result, a separate internal compartment is made, which can then fuse with other types of cellular compartments called liposomes. The digestive enzymes present in liposomes and finally destroy pathogens by breaking them down into bits. This is a picture of a non-specific innate response. Specific adaptive response, which is the third line of defence, aims to get rid of specific pathogens that have been encountered by the immune system previously. They can be adaptive or acquired immune response. Instead of being restricted to the site of infection, the adaptive immune response occurs within the body, throughout the body. And this goes on to say that the antibodies mediated immunity B cells start when they encounter a known antigen, and then they swallow and digest the antigen, followed by representation of the MHC. The combination of MHC activates a helper T cells, which the cytokines and interleukins, to trigger the growth of maturation and antigen-presenting B cells into the body, producing plasma cells and B cells and made memory cells, and keep the immune system ready for its next attack. Antibodies made by plasma are secreted into the bloodstream, where they start their functions in different ways by forming antigen-antibody complex. Antibodies can prevent antigens by binding host cells, leading to the prevention of infection. The antigen bodies complex can start a series of signaling events that activate the compartment proteins, which then kills pathogens by rupturing the cell membrane compartment, and proteins then trigger inflammation response, leading to an addition of white blood cells at the site of infection. In cell-mediated immunity, T cells start when they encounter antigen-preventing cells, such as B cells and dendritic cells. Activated T cells make cytokines, then they trigger production of maturation of T cells. Adult T cells grow into cytotoxic killer T cells, mainly destroying pathogen-infected cells, damaged cells and cancer cells, and rupturing the membranes. Most T cells mature into regulatory T cells and help stop the immune response by maintaining the immune system homeostasis. When a threat is eliminated, some T cells mature into memory T cells and remember the pathogen and start the immediate response when the body encounters a pathogen for the second time. Self-recognition, tolerance and description of specific immune system disorders. B-cell tolerance. B cells grow specific antibodies in the bone marrow, and the bone marrow contains pathogenic material and is full of antigens. If it develops into a B cell, it creates antibodies and match antigens and the bone marrow environment. One of two things happen: clonal deletion and clonal energy. These act as a means of testing B cells before they are restricted, released to ensure that they don't contain antibodies and that they are active against self. T-cell tolerance. T-cell tolerance leaves T cells leave the bone marrow as parathymocytes and travel to the thymus. When they become thymocytes, these thymocytes undergo a complex process to develop their TCR. Then they mature into CD4 and CD8 cells in the thymus gland. The specific for foreign antigen and self-MHC. If they recognize an antigen, they may cause autoimmune disease because they can't recognize self-MHC molecules. They are useless T cells. As useless as T cells. Dendrites begin by arranging proteins from the random TCRs. The first step stops expressing one of these, so they become either CD4 or CD8 cells. The second step meets crucial epithelium cells in the thymus, and they present with MHC proteins and thymocytes. Recognize the MHC protein, they can't recognize it and they're destroyed. Thirdly, they meet dendritic cells in the thymus and present them with self-antigens. Recognize the self-antigens and are destroyed, and they recognize it. When they can't recognize it, they allow it to pass through the thymus and become a fully fledged T cell. Autoimmune diseases. Diseases occur when the components of a specific immune system start to recognize self-antigens, leading to immune responses relating to self-antigen. They recognized and this result varies, and here are some examples. The autoimmune diseases can be type 1 diabetes, Graves' disease, systemic lupus erythematosus, myasthenia gravis, pemphigosis, Goodpasture syndrome, autoimmune thrombocytopenia, pernicious anemia, and automatic hemolytic disease, anemia. Section 3, acquired immunity, including a natural and artificial and passive, which including natural, artificial, passive and active. Immunity can start is a state of protection against infectious diseases, immune response generated by immunization or previous infection or non-immunological factors. This is a picture of active or passive immunity. Which is there. Artificially acquired passive immunity is immediate, short-term immunization provided by an injection of the antibodies isn't produced by the recipient cells. These antibodies are developed in another individual or animal, then injected into another individual. Anti-serum and a general term for this is for preparations that contain antibodies. Artificial active immunization is where the microbes or parts of it are injected into the person before they are able to take it in naturally. The whole microbes are used, and they're pre-treated vaccines, and the vaccine makes a primary response against the antigen in the person without causing symptoms of disease. Artificial passive immunization which normally is administered by injection and used if there has been a recent outbreak of a particular disease or an emergency treatment for toxicity, or like tetanus. The antibodies can be produced in animals called a serum therapy. They have a high chance of anaphylactic shock because the immunity against the animal serum itself. Humanized antibodies produced in a vitro by cell culture and are used and then it goes on to say the second artificial immunity in relation to the disease smallpox. Those individuals were exposed to a minor strain of smallpox in a controlled environment, and once their bodies had built up the natural immunity resistance to the weakened strain, they were much less likely to become infected with a more deadly strain. So the patients were given the disease in order to fight it in later life. And through this method was an effective one, although it was an effective one. The scientists of the time had no specific knowledge of why it worked. And this just goes on to say about the immune response and immunological, and how smallpox, the statistics about evaluated smallpox have been eradicated, and this is the concept of human herd immunity, and about different diseases that can be caused by infectious diseases there. And this is the references there.